Let's talk about nitrogen. Nitrogen's easier than carbon. Yeah, I know, it's like nitrogen is complex, but for animals it's pretty easy it's easier than carbon because, like I said, for carbon, animals can do things with the carbohydrates they eat, the lipids they eat, and the proteins they eat. All those things have different carbon isotope values, okay? Lipids are lower than protein, carbs are a little bit lower than protein, and they can scramble them up and use them in different ways to build these non-essential amino acids that we're measuring, essentially. That's not good. Non-essential amino acids that we're measuring, because most tissues are made of those non-essential amino acids. For nitrogen, it's all about protein. Carbohydrates don't have any nitrogen in them. Not a lot, at least. Okay, they have a little bit, but not a lot. Actually, they don't. I'm thinking about like cornmeal, which actually has amino acids in it. So they don't have nitrogen in it. And lipids only have a tiny bit of nitrogen in them, in the cell walls of lipids. Negligible amounts, okay? So really the only source here is the protein in the diet for nitrogen. Okay? Protein. It's all about protein for nitrogen. So again, we have to deal with assimilation, incorporation and routing of nitrogen, biosynthesis, and excretion. And when we're talking about excretion here, we're talking about losing light nitrogen, N14, in the form of different products, nitrogenous waste that we're all making. Nitrogen's actually, even though it's a limiting nutrient in most ecosystems, it's actually toxic for us in high quantities in our blood, and we have to constantly get rid of it. And we do that by peeing. And depending on whether you're a mammal or a bird or a lizard or a fish, you have different, basically, compounds that you're excreting nitrogen in, but nonetheless, you excrete the light isotope. Okay, so again, here's our protein cycling in mammals, amino acid pool. These three things come in, okay? Body protein, other end compounds. For nitrogen, in low dietary protein, you can have an essential amino acid deficiency. And this is basically what we're studying with the gut microbiome. What happens when animals aren't fed enough essential amino acids to do the things that they need to do to maintain their tissues? All right? And in high dietary protein, you can actually take that protein and you convert it to other things, glucose, like gluconeogenesis, and the excretion rates of these things go up. So actually, if you eat a big steak, and if you measure the urea concentrations in your pee, the morning after you eat a big steak, your urea concentrations will spike. Okay? Because your nitrogen, your protein intake just went up. So the nitrogen, you have to get rid of that nitrogen. Okay? All right. So here we go. This is pretty easy. Again, we're dealing with the amino acid pool. It's all about protein. This is the dietary protein that goes in here. You could potentially get some from body protein if you need it, but this is the protein pool. And through this processing here, you're losing the light nitrogen, which eventually you'll make into urea. Okay? And that's all done by these two non-essential amino acids in the TCA cycle. All right? Now we call that process by which an ammonia molecule, a light ammonia molecule in this case, splits off, we call that process deamination. Okay? So that goes back to your biochemistry textbooks. So you're constantly, when you're taking in nitrogen and you're making urea to get rid of it as a waste product, you're deaminating this ammonia off of it. And if you're a mammal, you take that ammonia and you build it and make it into a urea in the urea cycle. All right? But the deamination of the ammonia happens in all organisms. And that's the reason why you are what you eat plus 2 to 5 per mil. Again, we're talking about our glucogenic amino acids, there's our ketogenic amino acids. All the magic happens for nitrogen in this red box. That's all you need to know. You need to know this intermediary in the TCA cycle, and you need to know glutamate and glutamine. And then also pyruvate becomes important here. And I'll show you in a second why that is. But it's not as complicated as carbon. It all happens really right in this box. Okay? Glutamine and glutamate. Glutamine is the major amino acid that we use to shuttle nitrogen around to the body. Okay? So it's like the Santa Fe Northern. I don't know, the Delta Airlines. It's the carrier, right, of all the nitrogen that we need in various places in our brain, in our liver, and other organs. That's how we get it around, is via glutamine. Okay? All right, this is how it happens. So we have a protein. This is just an amino acid, right? An amino acid is a central carbon, an alpha carbon. It has a carboxyl, and it has an amine group. That's the definition of an amino acid. And then depending, this is the R group here, changes depending on what amino acid you are. But all aminos have that structure. Okay? And again, that can come from dietary protein, if you get enough protein in your diet, or if you're catabolizing your endogenous protein, it can come from your muscle stores. Okay? That gets transaminated. We take alpha-ketoglutarate, remember that's that important intermediary in the TCA cycle. We take this and we transaminate on that amino to alpha-ketoglutarate. Okay, and when we basically add these two molecules together in that transamination process, we create alpha-keto acids and glutamate. Okay? So we need this dietary protein, this amino basically, we need this amine group to make this process happen. Okay? Products, reactants. Pretty simple. And then we deaminate that glutamate to make alpha-ketoglutarate. Okay? So it basically just goes in a big circle. All right? So if you took this structure, you took that off of it, you get that structure, more or less. Glutamine can come in here, okay? And you can basically transform glutamine into glutamate. All right? And when you do that, you deaminate as well. Okay? So going back to this guy, as you're going this way, you're transaminating off, you're putting on an amino. When you're going this way, you're in this direction, you're doing deamination. Okay? All right? So as you're converting glutamine into glutamate, you're deaminating and you're creating more. This is the pool of ammonia that you're eventually going to either excrete directly if you're a fish or an invertebrate, or you're going to use to build urea and excrete as urea if you're a mammal, or uric acid if you're a bird. And there's one more important thing. Alanine comes in here. Remember, alanine is actually a glucogenic amino acid from glycolysis. But if you can deaminate alanine, that adds to this pool to make pyruvate. Okay? Alanine is a pretty common amino acid in diet. And if you need pyruvate and you have a lot of alanine, then you can convert alanine into pyruvate. And when you do that, you cleave off this nitrogen, and that adds to this pool as well. So that's basically how you do deamination to create the ammonia pool in your body that you eventually excrete. And again, that ammonia pool is 14NH3, by and large. Does that make sense? Questions? As someone pointed out, was it you, Will? Someone pointed out about directions, concentrations. Yes. So how fast this happens, whether this comes in, it all depends on the concentration of these different enzymes and these different materials in the body. Okay? You can actually take glutamate and you can go the other way if you wanted to and make glutamine, right? It just depends. It's a two-way street. The body is a big two-way street. And so concentrations really determine the direction of these things and the rate at which they happen. That's the cool thing about biochemistry. It's not as fixed as you might think. Okay? And again, like I said, you take that ammonia pool, and if you're a fancy mammal, you do urea. Cool thing is a lot of mammals, like bears, for example, they can take this urea, and when they hibernate, they don't pee it out. Why lose that nitrogen? They keep it and they recycle it. So they basically become like big nitrogen recyclers. Really cool stuff. Animals that hibernate, they can do this very efficiently.
Basically, this could affect their delta 15.
It could affect their delta 15N, yep. Because if you're not peeing out the light stuff, you're not going to be enriched. Okay, so you might expect a difference in their discrimination during times of hibernation. Okay. Nitrogen excretion influences. So if you look at animals that produce urea, like mammals, or animals that produce uric acid, like birds and a lot of reptiles, animals that produce ammonia, okay, like many invertebrates and fish, all right? The discrimination here, this is nitrogen isotope values of your tissue versus your diet, right? It varies pretty systematically. Mammals and birds have fairly high discrimination factors in comparison to the lesser organisms, the invertebrates and the fish that excrete ammonia. Okay, and that's about a per mil on average. There's about a per mil difference there. So you are what you eat plus 3 per mil in mammals and birds, but you are what you eat plus 2 per mil if you're a fish. Okay? And then you have these weird ones, things that like, not a lot of studies go here. See, there's a number of studies, 4 studies at this particular. This is published in 2003. One of my favorite actually papers. It's now a classic, but I think it's a really good paper by Vandercliff, Matt, and I don't know Ponsard's first name, but these are the weird ones. Like, why would you ever excrete amino acids? Are you nuts? It's like gold, and you just like, it's like a billionaire strategy, you know? What? It's like, doesn't make any sense, right? But if you eat a lot of protein, why not, right? So there are some insects that actually do this, which is pretty crazy. Okay? But for the most part, it does systematically vary depending on what excretion pathway you're using. So that's another important point to keep in mind. Nitrogen isotope discrimination also varies with dietary protein content. Okay? So this is dietary protein. We're not talking about quality. We're just talking about amount here. All right? And this is nitrogen isotope discrimination. You can see under low dietary protein contents, there's low discrimination. There's no discrimination between you and your food. Why? That's right. You can't be picky. Yeah. Why? You can't discriminate. If you need all the nitrogen that comes in, you can't get rid of it. You can't like say, oh, I'm only going to take the 15 in and leave the 14 in. I just need it all, so I just take it all. Can't be picky when you're poor. Okay, nitrogen poor. All right, but when you have plenty of protein in your diet, you can be picky. All right, and you can discriminate. So you can start shelling out that lighter isotope, and as a consequence, your body tissues become enriched, and your discrimination factor gets bigger. Okay, so that's an important point, an important concept. So pickiness, that's something that you can apply to a lot of things you've learned, like discrimination during photosynthesis. Who's more picky, C3 or C4 plants? C3 plants are more picky, yeah. They incorporate more of the 12C into their tissues, hence they have lower carbon isotope values. They discriminate more against the 13C. C4 plants don't care as much, okay? They take in more 13C. That's why they have higher carbon isotope values. Because again, carbon isotope values are just a measure of the ratio of 13 over 12.
What animal is this?
That's a fish. Yeah. But you see the same thing, and we've done experiments on mice. You see the same things across. This is a tilapia.
Same trend.
The same trend. There's actually 2 competing hypotheses. It has to do with nitrogen balance. And there's positive nitrogen balance. There's neutral nitrogen balance, and then there's negative nitrogen balance. And in negative nitrogen balance, what happens is you start eating yourself. These fish were probably in neutral nitrogen balance because they still grew a lot during this experiment, okay? But if you're in negative nitrogen balance, like my wife was during lactation, well, actually she wasn't, sorry, that's a different thing. What happens is you catabolize you basically go back up. So you might expect something like this to happen. Whoa, this to happen, like that. As this, if you change this axis to nitrogen balance. Okay? All right, another reason why nitrogen isotopes vary is protein quality. So the last graph was protein content, protein quality. What do I mean by protein quality? I mentioned it before, but let's recap. Kind of, non-essential versus essential, kind of, but someone else said something? It's basically how the match of your diet matches your needs. So if you need a lot of threonine and your diet has a lot of threonine in it, you have a high protein quality. If you need a lot of threonine and your diet doesn't have a lot of threonine in it, it's a low protein quality. So there's a way you can do that. It's called the biological value. You can estimate that. You can just basically match. You can look at the distribution and concentrations of amino acids in the diet versus the concentrations of amino acids that you need to do what you need to do. And if there's a mismatch there, it's a low protein quality. If they're perfectly matched, you have high protein quality. Okay? Look at this graph. This is cool. This is by Charlie Robbins' group up in Washington State, who's like legendary bear isotope ecologist. Here's animals that are raised on different things. Fruit, look at that. Look at the discrimination in animals that eat fruit. This is why I don't eat fruit. It's bad for you. It doesn't have any nitrogen, okay? You have to, okay? You wouldn't expect from the last graph, because it doesn't have any nitrogen, for like this to be actually down to 0. That's what I thought, but this is the pattern that they found, okay? But this high discrimination value suggests that there's some sort of recycling going on in the animal, and the animal's working extra hard in order to get its nitrogen balance. All right, but I don't eat fruit, and this is the reason why. Ruminant diets, animals that eat pretty crappy diets because they have the digestive physiology to prove it. Other animals, fish, everyone says eat fish, it's good for you. Well, there's some evidence to show that. Pretty high biological value of fish. And as you increase the biological value, you get this nice trend. Look at this, animals that eat milk diets, i.e. babies. Mom knows best. Evolution has driven biological value, the protein quality of milk matches perfectly what baby needs. Makes total sense. Mom knows best.
I thought that when you had a low protein diet, you had less discrimination.
I'm just showing you. So this is biological value. The last one was protein content. This says nothing about whether or not this protein was good for the tilapia. Okay? This protein in this experiment was actually casein, which is a milk protein from a mammal, from a cow. So why would you feed milk protein to a fish? Well, it's cheap. It has a different isotopic composition than the other dietary ingredients, and that's why they did that. Okay, so these are two different kind of concepts. It's a good catch, though. Okay? So you see this decrease in discrimination as biological value increases, okay? Suggesting that there's less recycling of nitrogen and things are getting more just basically routed straight in when the isotopic or the amino acid composition of your diet matches that of your needs. Okay. All right, I'm going to stop there. And the take-home messages for this, I can talk a little bit more if you want about gut microbiomes because I have a couple slides, but the take-home message is an animal's isotopic composition mirrors that of its food, but it's offset by predictable amounts, and we call those predictable amounts trophic discrimination factors. And we need to understand what those are and how they vary if we're going to apply them to wild animal populations. We're going to use them to study what bears eat in the wild, okay? What kangaroo rats eat in the wild, what sea otters eat in the wild, okay? So here the point is, don't poo-poo dietary experiments. Turi showed you this morning how useful they can be. A lot of that data was generated at the farm, literally. Okay? A lot of the data in our lab is generated in the basement of our building by feeding animals different stuff. All right? And those experiments can be incredibly productive, and you can learn a lot. I had an undergrad last year who did an experiment. He's working on his 5th paper from that one experiment as an undergrad. It's pretty awesome. So don't poo-poo these controlled feeding experiments. And if you work on animals, or plants for that matter, consider putting one of these into your dissertation. Okay? They're productive. Carbon isotopes is complicated because different dietary macromolecules can have very different carbon isotope values. Lipids is the primary example. Their carbon isotope values of lipids can be 6 to 8 per mil lower than proteins. Okay? So the relative influence of these different dietary macromolecules in non-essential amino acid synthesis can impact the discrimination factor. All right, so non-essential amino acids really drive the bus in terms of proportion of amino acids in a tissue. And if you can make them from a wide variety of things, you have to consider those other things in the diet of the animal in order to better understand what the carbon isotope discrimination is of that animal. Nitrogen, consumers have higher nitrogen isotope values than our food. They retain 15N, they excrete the 14N. 14N, we talked a lot about this via ammonia, urea, and uric acid. 15N varies with dietary protein content, protein quality, and catabolism. 15N increases positively with dietary protein content. I showed you that. 15N decreases with increasing dietary protein quality. And 15N increases with increasing catabolism of endogenous protein. I didn't show you that last little figure. That's from a paper that we just submitted. But we talked about it quite a bit with the use of endogenous stores and increasing your nitrogen isotope value as a function of catabolism. So the interpretation of isotope data relies on knowing which factors control the assimilation, synthesis, and as Terry mentioned this morning, the incorporation of isotopes into tissues. All right? And really one of the best ways at getting at this stuff from a physiological and biochemical perspective is using feeding experiments in animals in the laboratory or the zoo to understand what's better going on, because you can control those conditions a lot easier. Okay? People that work on plants do the same thing in greenhouses, right? All right, so that's it. I'll let you guys go.